All right, so the last part of this chapter is all about the organic molecules of life, which are your carbohydrates, your fats, your proteins, and your nucleic acids. And again, organic molecules are carbon and hydrogen containing. That's what this came from. So I like to start this off with a song. I love this song. It's one of my favorites, and I did tell you that I would be playing a song. If for some reason the song doesn't play well, all you have to do is open your lecture PowerPoint up online, so you have to be connected to an internet, and then click on the organic molecules here, and it will bring you to this wonderful song that talks about all of the organic molecules. And hopefully you will enjoy this a lot, because I know I do every time I hear it, and I've heard it a lot. It's like one of my favorites. Of course, most of my songs are also one of my favorite songs, just the way it is, right? Okay, so hold on. I want to make sure, hold on, I want to make sure that my volume is up. Okay. All right, ready?
Molecules gone wild.
Biostyle.
Carbohydrates provide the energy for your life. The simplest form, like glucose, is known as a monosaccharide. Combine two and make a larger sugar called disaccharide. The largest is a polysaccharide. Lipids are the fats. Decided gases, triglycerol makes up the fats. Triglycerides is only single bond unsaturated fats. But if they double bonds, they now become unsaturated fats. Don't eat too much fat. Monomers build to polymers, build with CHO. Molecules that grow. One with HO, other with HO. Bond and co. Research to O. I can't just because this is underway. Molecules gone wild. Biostyle. Molecules gone wild. Biostyle. Molecules gone wild. Many polymers. Molecules gone wild. Many polymers. Proteins are building blocks that make up your whole body. Your skin, your hair, your muscles need proteins to get the hottest. Break down polypeptides to amino acids in your body. Nitrogen comes to the party. Nucleic acids. This one is known as a... Oops, I made a mistake, sorry. Which makes into single nucleotides and is digested. Phosphorus added. When you eat food to get down, the flow. Straight down your throat. Need to break through the polymers. So here's what you know. Hydrolysis is what I just understand.
Molecules gone wild.
Molecules gone wild. Biostyle. Molecules gone wild. Biostyle. Molecules gone wild. Many polymers. Molecules gone wild. Many polymers. Food deforms into its simplest forms. When you eat it, break it down, then it reforms.
Now you're informed, so please study more. Study, take your notes and text if you know what I'm saying.
Molecules gone wild.
Many polymers.
Molecules gone wild. Many polymers. Molecules gone wild.
Okay, so super fun song to bring us into the last section of this chapter. So we're going to go through each of these organic macromolecules of life. We'll talk about the small monomers that make them up. And we'll talk about their major functions, as well as some of the most important molecules in each of the groups. So let's start with what a macromolecule is. A macromolecule and a polymer are the same thing. They're very large organic molecules. So when I say macromolecules, I'm talking about carbohydrates or fats or proteins that are made up of identical or similar subunits. So your carbohydrates are made up of small monomers known as monosaccharides. Your proteins are made up of monomers called amino acids. Your nucleic acids, macromolecule again, are made up, like DNA, are made up of nucleotides. And your lipids are made up of small monomers known as fatty acids and glycerols. So the first one we'll talk about is carbohydrate. So carbohydrates' major function is for energy. So it's for fast energy. Carbohydrates, specifically glucose, are oxidized to produce ATP. ATP is the energy currency that all of our cells use. The most important of all of the monosaccharides is glucose. Glucose is our blood sugar, and it helps us maintain normal energy function all day long. So here are the 5 monosaccharides that we can find in our body. Glucose, again, blood glucose levels, so very important. Fructose, glucose and fructose together make sucrose. That's your table sugar, so fructose is a sweet sugar. Galactose is milk sugar, is part of milk sugar. So when you have glucose and galactose together, you form lactose, and lactose is your milk sugar. And then we have deoxyribose and ribose, which make up part of your nucleic acids. So I just talked about glucose, fructose, and galactose, as well as deoxyribose and ribose. Sucrose is formed when you take glucose and fructose and put them together. So that's this molecule right here. Sucrose is table sugar. Lactose is glucose plus galactose. And then maltose is grain sugar, and that's like the what's in wheat, potatoes, well, not so much potatoes, well, yeah, potatoes, I'll say that. Glucose and glucose, okay? So I guess your wheat, your oats, barley, stuff like that would be maltose, okay? And then we have important polysaccharides. So when you take monosaccharides and put them together to form disaccharides, this is 2 single monosaccharides put together through dehydration synthesis, forming the disaccharides. When you take multiple monosaccharides and you put them together, then you form polysaccharides. The polysaccharides are the macromolecules or the polymers of the carbohydrate world. So the important ones that we have are starch, glycogen, and cellulose. Glycogen is the storage form of glucose in our cells. So we use glycogen in our cells to store glucose until we need it. So that, like, if you have a big meal and you have extra sugar in your blood, you're going to have glucagon come out, and glucagon will tell your cells to take in glucose and build up this glycogen molecule. And then when we need glycogen or glucose in our blood, we can break the glycogen down. Starch and cellulose are plant storage forms. So starch is the storage form of glucose in your plants, just like glycogen. And then cellulose is the structural form of glucose that's found in plant cell walls. We can't break cellulose down, so it becomes the fiber in our diet, so it's still very important to us. The next organic molecule is lipid, or fats. Lipids are hydrophobic, whereas I did not say this before, so I'll say it now. Carbohydrates are hydrophilic molecules. They will break down in water, whereas lipids won't. If you've ever taken oil and mixed it with water, oil does not mix with water. It stays separated from the water. Lipids are hydrophobic molecules. They are not as easily oxidized as carbohydrates, and so they end up having more calories to them. They are also used for energy, but for long-term energy storage. So we can store them for long periods of time and then use them when we need them. Fats are used for a lot of different functions, which I'll talk about in a little bit. So first, let's go over the 5 typical fats that we find. Fatty acids, which make up all of our fats pretty much. Triglycerides, phospholipids, eicosanoids, and steroids. So eicosanoids, an example that your textbook likes to talk about or does talk about, is prostaglandins. The primary function of fat is for energy storage, insulation, so that's what this is showing, and shock absorption. Fatty acids are composed of long chains, carbon chains, that are either saturated with hydrogen or unsaturated. So if it's saturated with hydrogen, then it's considered a saturated fat, and this is an animal fat. If it's unsaturated, then it is an unsaturated fat, so it's not saturated with hydrogen. Here, there's a single bond, so this would be a monounsaturated fat, and that's usually a plant fat. It could also be with multiple unsaturated regions, and that would mean that this would be a polyunsaturated fat. The first real molecule that we talk about is triglycerides. Triglycerides are fats that are used for that we take in, used for energy or to help with building up other molecules or with protection or warmth, or so all the different functions that fats give. Triglycerides have 3 fatty acid chains, and they're linked to glycerol via dehydration synthesis. And I will say, if the triglyceride is liquid at room temperature, then it's a plant triglyceride. If it is solid at room temperature, then it is an animal triglyceride. Phospholipids. So up here you see a phospholipid. Phospholipids have 2 fatty acid tails linked to, again, glycerol, and they contain a phosphate group. So this is a phospholipid. Phospholipids are what make up the plasma membrane of our cell, the majority of the plasma membrane of our cell, so very important because our cells need that plasma membrane. We have sterols, so cholesterol is an example. Steroid molecules are all based off of cholesterol, which is a sterol. So this is what cholesterol looks like. Your testosterone, your estrogen, cortisone, they're all sterol molecules based from the cholesterol molecule, which is produced in the liver. And then we have our eicosanoids, specifically prostaglandins are a specific type of eicosanoid. Prostaglandins are localized hormones that respond to changes in the environment, and they can cause inflammatory responses. They can also make you more susceptible to pain reactions so that you feel the pain more. And so those are our fats. And now we're going to get into proteins, probably the most discussed of our organic molecules here. So all proteins are composed of amino acids. Amino acids all look identical except for this R chain or this side chain down here. So they have an amine portion, they have a carboxyl portion, they have an individual hydrogen on a carbon, and then they have this R chain. This R chain is what changes in each amino acid. There are 20 different amino acids that make up every protein that makes our bodies up. And proteins make up everything. Hair, skin, nails, teeth, blood, everything that you see, everything that allows you to move is protein. Proteins are formed when amino acids bind through dehydration synthesis and produce peptide bonds. So a single amino acid peptide bond would form a dimer of amino acids, but when you get multiple peptide bonds, like 50 or more, that's when you end up with a polypeptide, and a polypeptide is the primary form of a protein. And so here you can see the four types of protein folding. So the primary structure of a protein, and I will say this really quick before I go over the four structures. With proteins, the structure is what determines how it functions. Remember, structure determines function. For proteins, the amino acid sequence determines its structure, and then that will determine how it functions. So if you have an amino acid sequence that is not correct, then it's not going to have the proper structure, it won't have the proper function. So the primary structure of a protein is the amino acid sequence. This is known as the polypeptide. So here's amino acid 1, 2, 3, 4, 5, yada, 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 all the way through amino acid 850 or whatever. That's the primary form of the protein. The amino acids start to fold because you have these R groups, and these R groups may have a positive or a negative or a neutral charge. If you have positive here and a negative here, they might become attracted to each other, forming a pole or a bond. These bonds might be hydrogen or ionic bonds that help to make the protein form in either alpha helix or beta pleated sheet. That is the secondary structure of a protein. From there, it folds into its conformational form, which is the tertiary structure. Tertiary structure is the functional form of a protein, but some proteins have a quaternary structure. A quaternary structure occurs when you have multiple tertiary forms come together. So basically you have maybe 2 or more polypeptides that fold together, forming a large quaternary protein. So the best example in our body is this, which is hemoglobin. Hemoglobin is composed of 4 individual polypeptides that form their tertiary structure all together. And then they have attached to them this heme group, or this heme moiety, which is not a protein but is important for the function of this molecule. So the shape of the protein, or the conformation of the protein, is going to dictate its function. Structure determines function. And conformation, so if the conformation is wrong, then the function is going to be wrong. And if you change the conformation, then that's going to destroy its function. So denaturation is a conformational change that is unchangeable. So if the protein changes in shape and cannot go back to its original shape, that is known as denaturation. Typical reasons for denaturation: extreme heat or a change in pH, either lower or higher than it's supposed to be. So in these cases, when you have this change, the function becomes nil. So think about when you cook an egg. The outer liquid, kind of mucousy clear material, that's called albumin. And when you put it on a frying pan and you fry it, it turns white. It has changed in conformation, and it no longer would have the same function. It becomes a rubbery material and would no longer be able to do its normal job. That is denaturation. So proteins have multiple functions. They have structural, they have communication, transport functions, movement functions, binding, and of course they are enzymes. So structurally, we have keratin that makes up our skin, our hair, our nails. Beautiful hair, by the way. I love this. I wish this was my hair. I used to have hair like this when I was in high school. But yeah, that's a total other story. I guess you don't care about that. Anyways, so keratin is a structural protein. Over here we have bone showing. Bone is composed of collagen proteins. Collagen is another structural protein, and it's also a very hard protein that is very protective. We have some proteins that are communication proteins. So right here we have a glycoprotein. Glycoproteins act as communication molecules. So they can interact with other molecules and they can bind other molecules and allow for interaction between them. We have membrane transport proteins. So integral proteins. So here's an example of an integral protein. Known as a transport protein that allows molecules to move across the plasma membrane. They can't go through, they go right across. We have some molecules, or some proteins act as just movement. So proteins in our muscles allow us to move. These same types of proteins can also move along the cytoskeleton of the cell and move materials from one area of the cell to another area of the cell. Binding. We have proteins that bind cells together. So here are two cells, and we have these different proteins, not so much gap junctions, but these different proteins, adherins and desmosomes, that link the protein, the plasma membranes together, and tight junctions up here. Or down here, we also have proteins binding the cell to the basement membrane of the cell. So basement membrane is composed of some type of other tissue, like connective tissue. Okay. And then we have proteins that act as enzymes or catalyze reactions. And so here we have two different substrates. So these are the reactants. They bind into the active site of the enzyme. The enzyme gives a little squeeze and produces a chemical reaction that, in this case, binds these two substrates and forms one product. So this would be an anabolic and an endergonic reaction. Enzymes speed up the rate of a reaction by reducing the amount of energy it takes for the enzyme or for the reaction to occur. The last molecules that we'll talk about that are organic are nucleic acids. Nucleic acids are primarily your DNA and RNA. They're composed of nitrogenous bases, either adenine or guanine, which are your purines, or cytosine, thymine, or uracil, which are your pyrimidines. They contain a phosphate group, and they contain a sugar, either deoxyribose or ribose. This is DNA, and over here you can see the 4 nitrogenous bases that are found in DNA. You can see that adenine always binds to thymine, so you have a purine and pyrimidine always binding. You have guanine always binding to cytosine, another purine and pyrimidine always binding. These produce 2 bonds, these produce 3 bonds between them, and they're held together by hydrogen bonds, whereas the backbone here, sugar phosphate backbone, is deoxyribose and phosphate, and that's covalently bonded through phosphodiester bonds. DNA carries the genetic information of you in each individual cell. DNA is what codes for every single protein that makes you who you are, and that DNA is what is transferred from parent to offspring. RNA then is the worker nucleic acid. RNA contains adenine, It's always single-stranded, so it doesn't bind to other molecules. It doesn't have a second strand. So adenine, guanine, and cytosine are found in your RNA, but instead of thymine, we have uracil taking its place. RNA is what takes and carries out the instructions of DNA in protein synthesis. So it produces the proteins. There are 3 types of RNA. We have messenger RNA, ribosomal RNA, and transfer RNA. And then we have one nucleotide that I really like to talk about, and this is called adenosine triphosphate, which is composed of an adenine, a ribose, and 3 phosphate groups. ATP is the energy currency that all of our body cells use. And it stores energy, a high energy bond between the 2nd and 3rd phosphate. So when we break that bond, that release of energy allows us to do work. This is just a figure from your textbook showing what DNA looks like. And guess what? That means I'm done with this chapter. So at this point, you should be able to go through all of the questions in the back of your chapter 2 and answer them. If you have questions, definitely ask. Bye.